Okay, morning. So what we're going to do today, going to review the carbohydrate metabolism materials. And again, I already mentioned the first 1 hour I'm going to go through some of the slides that I think is maybe kind of a little bit, complicated, try to clarify. And then the rest of the 1 hour that you guys can ask questions, right? Okay, so for the carbohydrate metabolism 1 and 2, I think the glycolysis, 5 pathways, like for glucose metabolism, glycolysis, glycogen synthesis, gluconeogenesis, and glycogen breakdown, and pentophosphate pathways, right? So 5 metabolic pathways, and I think the most, I will not say the most important, the important pathway that you guys want to probably to know the glycolysis, gluconeogenesis, glycogen metabolism, right? And pentophosphate pathway relatively simple, so I would suggest you focusing on more on this 4 main metabolic pathways, right? And glucose homeostasis. We body, just try to maintain blood glucose levels. So we have a regulatory system to, to make sure that we have right levels of glucose, either in fat condition or fasting condition. So, and this slide just showing in the fat condition, our body deal with the high blood glucose level, right? So try to reduce the glucose level after meal and to the normal range. So you see the distribution of the glucose that takes up from the diet, and then 20% of the glucose is metabolized in the liver, and you need to know which metabolic pathway that we talk about, is activated in the fat condition. Should be glycolysis or glycogen synthesis, right? So these 2 metabolic pathways is activated, and also the glucose uptake into the different tissues is also very important mechanism to reduce the blood glucose level and to metabolize glucose in the fat condition. So, and different tissues has the glucose transporters, right? Glucose, enter the tissue cells, need the transporter. And 2 main transporters that involve the glucose homeostasis or blood glucose control are the group 2 and group 4. I would like you guys to remember. So group 2 is glucose dependent, right? The glucose regulated by glucose level. So means that when glucose level is high, then group 2 is activated. And 2 tissues you also need to remember, right? Have a high levels of group 2, which are the liver and beta cell, right? Pancreas. And the other important transporter group 4 is insulin dependent, right? So regulated by insulin. And 2 main tissues, 2 main tissues, muscle and adipose tissue, are the group 4, the major expression group 4, and regulate by insulin. So, and the first TL response for the after meal is the insulin release, right? When you raise blood glucose level, and glucose goes into the beta cell and triggers insulin release, and you we have a test. We have a question in quiz 1, right? See, the intracellular events, which happen first, and the sequence of events in response to glucose to stimulate insulin release. Remember the GLU2 transport glucose into the beta cells and go to glycolysis and produce ATP. So I probably just go slide by slide, right? We'll talk about later the intracellular events of insulin secretion. So maintain the blood glucose level and after meal, during the fasting. So during the fasting, very important, the glucose store, the glycogen, right, store in the liver, and also store in the muscle. But muscle does not contribute in the glucose, blood glucose, because of a lack of enzyme, right, you guys? No glucose 6-phosphatase. So in the fasting, the major contribution to the glucose level is from the glycogen stored in the liver, and also liver, kidney can make glucose, right? Gluconeogenic pathway. So in other words, during the fasting, glycogen breakdown and gluconeogenic pathway are activated, right? And this I already mentioned, glucose transporters is very important to regulate the glucose into the cells and reduce blood glucose levels. So, and you just wanted to focusing on these two transporters, GLU2 and GLU4. One is independent, right? The other one is glucose level regulated. And this, again, the five, so all the transporters that you wanted to focus on too for the blood glucose control. And you also remember, right, the GLU5 SGLT1 are the glucose transporter that plays a role in the glucose absorption, right, in the enterocytes. So GLU5 basically transported fructose, and SGLT1 is a transporter glucose from intestine lumen into the enterocytes. And GLU2, remember, is also the glucose transporter in the basolateral membrane, right, on the of the enterocytes, and transporter, glucose from enterocytes into the portal vein. And how glucose transporter regulate glucose metabolism, basically just, I talked about already, two major transporters, GLU2, GLU4, and the different distribution, right, the different tissue distribution you need to know, and how they regulate, one by glucose and other by insulin, right? And this would be the how dietary glucose after meal that stimulate the glucose insulin secretion, right? So basically, in the fat condition, this GLU2 is responsible for glucose levels, and the two major tissues distributed in two major tissues, liver and beta cells, and they have a different functions, right? And in the liver, transport the glucose from circulation to the liver for glycogen synthesis, right, and stored as energy. And in the pancreas, GLU2 transport the glucose and to stimulate insulin release, right, to as the so-called first here response to blood glucose levels. So here is the beta cells. You wanted to know how glucose stimulate insulin release, and first the uptake by GLU2, and then go to glycolysis and raise the ATP to ADP ratio, and then depolarization of membrane because of potassium channel is open up, so potassium go out of cells and change the membrane polarization. And then the next step will be the calcium channel is open, so calcium influx into cell raise the calcium concentration inside of cells. And this increased calcium concentration triggers the movement of a secretory granule of insulin and release insulin, right? So just try to remember the the sequential events, which occurs first and which the next. So the first tier response is just respond to the high levels of glucose and triggers insulin release. And then once the insulin levels increase, and insulin stimulate GLUT4 and to uptake glucose in two major tissues, skeletal muscle and adipose tissue, right? So the first tier response involved in both beta cells and liver, right, and GLUT2, and second tier response involves the GLUT4, insulin GLUT4, and skeletal muscle and adipose tissue, right? And this you just get some idea, GLUT4 in the low level insulin, usually it stays inside of cells. And only when insulin stimulate, the insulin levels goes high and insulin stimulate translocation of GLUT4 from inside of cell to the cell surface and take up glucose. So this process is regulated by insulin, and in both adipose cell and the skeletal muscle cell. And in addition to insulin stimulation, GLUT4 translocation, skeletal muscle also respond to exercise. And exercise is also able to stimulate GLUT4 translocation to cell surface and uptake glucose. And this process is independent of insulin, right? Just respond to exercise through the different mechanism we're going to talk about very later. So, and then, it's just the, skeletal muscle, respond to exercise and also translocate, GLUT4. Okay, so, tissue distribution, we already talked about it. And you also wanted to know that different tissues metabolize glucose differently, right? Now I think you guys should have a better understanding, right? Like in liver, is most important tissue for glucose metabolism because it does all 5 different metabolic pathways, like glucoglycolysis, gluconeogenesis, glycogen breakdown, glycogen synthesis, and pentophosphate pathways. So, and you also want to probably know, the diagram to show the different metabolic pathways, be able to know which tissues or cells you can, recognize. This cell could be liver based on, the metabolic pathways in the happen or occur in this particular cells, right? And the skeletal muscle, you can see, can do the glycogen synthesis, glycolysis, and release the lactate, right, for gluconeogenesis in the liver. And red blood cells, because of lack of mitochondria, so only does the glycolysis, but also be able to do, the pentophosphate pathway. And good red blood cells also contribute lactate, right, to the circulation for gluconeogenesis in the liver. And glycolytic pathways. Glycolytic pathways, the 2 stages. You guys wanted to know the key reactions, right? 3 key reactions and key enzymes in the glycolytic pathways. And first, the glucokinase or hexokinase catalyze phosphorylation glucose. And the second key pathway, key reaction is the key enzyme is the phosphofructose 1 kinase, right? Catalyze conversion fructose 6 phosphate to fructose 1 phosphate. And the first stage utilizes 2 ATPs and generating the 3 carbons, right? So the 6 carbon glucose molecules break down to 2 3 carbons molecules, right? Glycerol 3, not glycerol 3, glyceraldehyde 3 phosphate, right? And then these 2 3 carbon molecules then enter the second phase. The second phase basically is the stage that's generating ATPs and NADH, right? So 2 ATPs generated at the substrate phosphorylation levels, and because of 2 carbon molecules enter the second stage, right? So usually generate 4 ATPs at the second phase and 2 NADH molecules, right? And also depend on the metabolic stage, whether it's the aerobic or anaerobic conditions. So under the aerobic conditions, that the pyruvate will be the final product of glycolysis, and pyruvate will be further be metabolized in the mitochondria. So you remain the 2 molecules of NADH that will eventually enter the mitochondria for ATP production, right, and in the electron transport chain. And if under the anaerobic conditions, for example, in the red blood cells, because no mitochondria, so it cannot be further metabolized pyruvate, or under the the short-term exercise, and we will talk about later. So because of no oxygen, so under this kind of anaerobic conditions, the pyruvate will be further metabolized to lactate. So lactate is the final product of the glycolysis under the anaerobic conditions. And the NADH, because generated here but used up here, right? So you will not have any NADH molecules produced under the anaerobic condition, right? And okay, what is this? Oh, okay. So this is just a further, metabolize how pyruvate being further metabolized under the aerobic conditions. So pyruvate is generated through the glycolysis in cytosol and transported into mitochondria. We then break down to the acetyl-CoA molecules. Again, acetyl-CoA is very important essential molecules, right? Can generate it from oxidation of 3 micronutrients, and it is also very important molecules that enter the TCA cycles, and for the depend on again which type of cells. If it is in the liver, then the acetyl-CoA being condensed together with oxaloacetate to citrate, and citrate leaves the TCA cycles and go back to the cytosol, right? We talk about lipogenesis, remember? And for fatty acid synthesis or lipogenesis pathways. And if under the fasting conditions and in the liver again, the acetyl-CoA will not enter the TCA cycles, but utilize for ketone body synthesis, right? But it's okay. I think that we have not talked about this yet, right? But just let you know, these key molecules can have a different fate, depend on the cells and depend on the metabolic conditions. And the other sugars like fructose and how fructose is metabolized, and you just wanted to know that fructokinase is a key enzyme to metabolize the fructose because it's highly expressed in the liver. So fructose is mostly metabolized in the liver, and also in other enzymes, the aldolase B, also very high. So these two enzymes are the key enzymes that can break down fructose, right, to the 2,3-carbon molecules. So and then this 3-carbon molecules and join the the glycolytic pathway. So the main thing is because they bypass the key reactions, key enzyme, the control step in here. So fructose can be metabolized without critical control of so then fructose can be metabolized. A lot of fructose can be metabolized and generate the glycerol 3-phosphate, right? And this, is the backbone for fatty acid synthesis. So, and this just gives a little bit more details, how fructose, high fructose consumption can lead to the fatty liver or obesity, because it's you will generate a lot of pyruvate acetyl-CoA because of this bypass, the rate-limiting reactions. So you have a lot of fructose can be metabolized to generate pyruvate acetyl-CoA and for fatty acid synthesis, right? And the other reason is because fructose will not stimulate insulin release, right? So insulin is an important hormone that can stimulate glycogen synthesis. So glucose once can, if it's metabolized, the other pathway can also be used. Particularly fructose 1-phosphate can be used for glycogen synthesis. So you can reduce the production of this the pyruvate and acetyl-CoA, right? When insulin level is high. So when we consume glucose, but fructose will not stimulate insulin release, so this pathway cannot, it's blocked. So you have even more go to this pathway for fructose metabolism. And the kind of disease, I guess, right? The fructose intolerance, the reason is the lack of this enzyme. And so the lacks the LSB. So LSB deficiency can cause in the fructose 1-phosphate accumulation or build up, and this is a strong inhibitor of a glycogen breakdown, right? So the patient of LSB deficient patient usually have a problem with the blood glucose maintain, particularly when they consume a high fructose. And this just to show you the this is the one key reactions. The pyruvate generated from glycolysis of glucose can be decarboxylate to acetyl-CoA, and also this reaction produces NADH, and there is kind of a negative regulation, means that if the product acetyl-CoA or NADH levels high, and these two substrate or products can negatively feedback, inhibits the pyruvate dehydrogenase activities, right? So negative regulation. And acetyl-CoA again is a key molecules and can be generated from metabolism of amino acids, fatty acids, and of course glucose. And it also can be used for fatty acid synthesis, ketone body synthesis, cholesterol synthesis, and also can be further complete oxidized in the TC cycles. And it's just the overall pictures. I think it's very important to connect all all the nutrient, 3 macronutrient metabolism to TCA cycles and to electron transport chain, right, for ATP production. And again, acetyl-CoA is a key molecules and generate from metabolism of carbohydrate, protein, lipids, and enter the TCA cycles and generating the ATPs and most importantly reducing equivalent NADH and FADH. And FADH and NADH eventually has to go to the electron transport chain for ATP production, right? So electron transport chain or oxidative phosphorylation process is the main function of this process is for ATP production from NADH and FADH, right? And TCA cycles, the main function of TCA cycle is for the oxidation and produce CO2, water, and produce the ATP and reducing equivalent. And the other important function of the TCA cycle is to connect the the metabolism, glucose and amino acids and fatty acids metabolism, right? And it's also very important TCA cycle that involve the gluconeogenesis particularly from amino acids, right? So amino acids, if amino acids are used at TCA cycles, and you will enter the TCA cycles and to convert it the oxaloacetate. So oxaloacetate is also the key molecules in the TCA cycle that connect the amino acids to glucose metabolism, right? So and lots of different amino acids can be enter TCA cycles and converted to oxaloacetate. So oxaloacetate leaves the TCA cycles and go to the gluconeogenic pathways, right? So if you remember the analog process and catalog process, the concept, right? So this two, okay, let me finish. So let's go to the next. That's the metabolism 3 and 4. Let's see. Okay, question? You mean, I'm sorry, the gluconeogenesis? I think it's a kind of mix. Yeah, I think it considers the kind of mix. I mean, because you first of all you break down the larger molecules to small molecules, right? And it's small molecules like lactate or pyruvate and amino acids that are used for synthesis of glucose, right? So the part is the anabolic. So the kind of mix involve catabolic and anabolic. Not very. I don't think it is this gluconeogenesis. It can be very clearly defined as catabolic or anabolic, right? Yeah, probably it's also similar like gluconeogenesis. The glycogen breakdown, glycogen synthesis, and glycolysis is pretty clear, right? The catabolic, anabolic. But this two parts, gluconeogenesis and PPP, I mean, was not so clear. But yeah, it's okay. And here, the gluconeogenesis, right, the whole process, just, glycolysis occurs in the cytosol and the TCA cycles in the mitochondria, and also oxidative phosphorylation also occurs in the mitochondria, right? So mitochondria is very important for doing the anaerobic metabolism, right? And electron transport chain, we have like 4, including ATP synthesis is actually 5 complex, and 3 complex, 1, 3, and 4 are the proton pump, right? Be able to generate proton gradient, and proton gradient contains energies. So once a proton can re-enter the mitochondria and then reduce the gradient, and then also release energy. This energy from the release from proton gradient can be transferred to ADP molecules and synthesize ATPs, right? So this is a diagram showing, and you also wanted to remember that NADH transfer electron to complex 1 and then 3 and 4. So again, these 3 complex are located in the inner mitochondrial membrane, so be able to generate proton gradient, right? So these 3 complex be able to release the energy because they're able to generate proton gradient, right? So able to release energy and for ATP production. So each complex will release energy that enough for making 1 ATPs, right? So that's the reason NADH usually will generate 3 ATPs, right? And FADH, they actually transfer electron to complex 2, because complex 2 is localized inside of mitochondria, so we're not able to generate proton gradient, right? So complex 2 will not have energy for ATP production. So actually FADH just transfer electron to complex 2 and then 3 and 4, so only generate a 2 proton gradient, right? So that's the reason FADH only produces 2 ATPs. And we also talk about the shadow system. Remember the NADH can be generated in cytosol from glycolysis, and mitochondrial membrane is not permeable for NADH molecules. So that means NADH needs a shadow system to transport its electron to the another reducing equivalent inside of mitochondria. So and glycerol 3-phosphate be able to transfer electron from NADH in the cytosol to FADH in the mitochondria. So it is FADH in the mitochondria that goes to the electron transport chain for ATP production. So this is the reason why we mentioned the NADH produced from glycolysis sometimes generate 3 ATP, sometimes generate 2, right? Just depend on which shadow system that transfer electron from cytosolic NADH to the other or different reducing equivalent. And the other shadow system is mainly aspartate shadows, right? And this shadow be able to transfer electron from NADH in the cytosol back to the NAD, so generate NADH in the mitochondria. So this system is able to produce the 3 ATPs, right, from cytosolic NADH. Now gluconeogenesis. So gluconeogenesis is the pathway that can produce glucose from non-glucose precursors. So precursors of gluconeogenesis can be glycerol, pyruvate, lactate, propionate acid, right, and also some amino acid that can be converted to oxaloacetate or pyruvate. And liver is the main tissue, right? Liver and kidney are the main tissue that be able to do the gluconeogenesis during the fasting. And the gluconeogenesis occur in both cellular compartment, the cytosol and the mitochondria, depend on the reactions. But it's not exactly reversal, right, of the glycolysis. Remember, glycolysis has three, the key pathways, key reactions, enzyme that cannot be converted, cannot be reversed. So in gluconeogenesis, it requires a different set of enzymes to catalyze the reversal reaction. So this is just the precursors, right? So so you try to remember the key reaction enzyme in the gluconeogenesis, right? Again, this is the overall pictures. The gluconeogenesis, the sum part of the two reactions occur in the mitochondria and the other two reactions in the cytosol, and also depend on the precursors. If precursors are the pyruvate or lactate, then it will go for reactions, right, in the mitochondria and the cytosol. And if precursor is the glutamate, amino acids, and you only have one reaction in the mitochondria and two reactions in the cytosol, right? Remember we will talk about this. And if glycerol as the precursors, this will only go to key reaction in the cytosol, right? And will convert it to the glucose. So it depends on, again, what type of precursor I used for gluconeogenesis. The reaction can be, a process can be different. And these are the two key reactions in the cytosol. You guys wanted to remember which enzyme or reaction in the glycolysis and which enzyme in the gluconeogenesis. And the last reaction is the conversion of glucose 6 phosphate to free glucose, and this is a key enzyme, glucose 6 phosphatase, right? And I mentioned that glucose 6 phosphate present in liver and kidney, but not in the skeletal muscle. So that's the reason, kidney muscle can do gluconeogenesis and be able to release glucose into the circulation, and muscle does not directly contribute the glucose into the circulation. And the two gluconeogenesis cycles, Cori cycle and alanine cycles. The Cori cycles, basically you get lactate from muscle and red blood cells and make the glucose in the liver, right, and then contribute glucose to the circulation. And alanine cycles, the pyruvate, the alanine is released from skeletal muscle and picked up by the liver, and for gluconeogenesis, right? So just see which substrate and which tissues contribute to the substrate for gluconeogenesis, and try to remember this process. And this I just mentioned that if some amino acids as the precursor to gluconeogenesis that involve some reactions in the TCA cycles, right? So main the process is the amino acids through the several reactions and converted to oxaloacetate, the key molecules, and then this leads the TCA cycle for gluconeogenesis, right? And you also know that the acetyl-CoA that connect to citrate and for fatty acid synthesis, lipogenesis, right? And the two clinical correlation, just wanted to you guys understand, this whole process. There's a key metabolic pathways, gluconeogenesis, all the metabolic pathway glycogen breakdown and gluconeogenesis. And the two key enzyme deficiency, what happens to blood glucose control and lactate levels and the glycogen store, and depend on the, particularly for glycogen store, for the glycogen, right, whether it's causing glycogen depletion or causing the glycogen accumulation, depend on the two different enzymes. And the reason I think that we, if the glucose 6-phosphatase is deficient, will also causing hypoglycemia, have a low glucose levels and higher lactate levels, but causing the glycogen accumulation. The reason, because remember, when we talk about glycogen metabolism, when the glucose 6-phosphatase is deficient, then glucose 6-phosphate cannot be converted glucose. So you have a glucose 6-phosphate build up. So glucose 6-phosphate is a strong inhibitor of glycogen breakdown, remember. So that's the reason why, deficient of a glucose 6-phosphatase causing the glycogen accumulation. And glucose neogenesis in the kidney basically is happened a little bit late compared to the liver, glycogen breakdown. So when the starvation goes longer, for example, longer than 1 day or 24 hours, and then kidney becomes an important gluconeogenic organs, right, and produce the glucose. And kidney, gluconeogenesis in kidney mostly use amino acids as the precursors, right, so a little bit different from the liver. And this process, catalysis, basically is the two opposite process that the main function of this is try to maintain the intermediate of the TC cycles, right? If we adding the intermediate into the TC cycles, this process called anaerosis, and catalysis, that means that we take some intermediates of TC cycles and to make either glucose or the fatty acids, right? So this process called catalysis. So basically, the anaerosis and catalysis just try to replenish the intermediate TC cycles and to prevent the buildup or the depletion of intermediate to make the TC cycle functional, right? So this is the main purpose of the anaerosis and catalysis, right? And yeah, I think I don't want to repeat this, but you guys just want to know which process is the catalysis and which are the anaerosis, right? Okay, so yeah, let's just say, the role of a TC cycle in the gluconeogenesis involve anaerosis and the catalysis process, right? Okay, pentophosphate pathways. So the substrate for this pathways is glucose 6-phosphate, right? And the purpose of this pathway is to produce the 5-carbon sugar phosphate, right? And also to generate NADPH. So NADPH is very important, the electron donors, right, for the fatty acid synthesis. Remember we talked about already, right? So pentophosphate pathway is one of the sources of NADPH for lipogenesis. And the regulation. So regulation, with the different levels, transporter levels, substrate levels, and the enzyme levels, right? And hormonal regulation. And the transporters I guess you probably wanted to understand we also introduced the two different concepts, right? Affinity for glucose and Km, which is opposite. So you probably want to understand why they are opposite, right? And which, glucose transporter has a high affinity or low Km or something like this, right? And yeah, the similar stuff. So we don't want to go into these details. And substrate regulations that you wanted to know that the fructose 1 phosphate, the fructose 6 phosphate, insulin, glucagon, epinephrine, citrate, and AMP, and how they regulate the different sets of key enzymes in the two different pathways under the different physiological conditions, right? And it's just the whole picture of how metabolism is regulated by hormones, nutrients, and enzyme activities and gene expressions, and how insulin regulates. Insulin basically increase the glycolysis and glycogen breakdown pathways, right? And so you can imagine, the insulin should stimulate key enzymes in the glycolysis and glycogen synthesis. And in the meantime, insulin should suppress or the glycogen breakdown pathway or suppress the gluconeogenesis. So it should inhibit the gluconeogenic enzyme activities and the glycogen breakdown enzymes, right? And so the main function of glucose is to reduce blood glucose level. And the other key enzyme, the key hormones that regulates the metabolism, glucagon and glucocorticoids and epinephrine. And the glucagon basically raised during the fasting condition, right? And should stimulate gluconeogenesis and glycogen breakdown, and should inhibit the glycogen synthesis and glycolysis, right? Okay, so I have actually one more set of slides. Do you guys want to take a break and then I finish then the one more set of slides, and then we open for a question, right? Okay, so and then the carbohydrate metabolism 5 and 6, um, mostly on the regulation of enzyme activities in glycolysis and gluconeogenesis pathways. Um, and I think focusing on the three key reactions, right, and the bypass reactions and key enzyme, how they regulated by substrate, the hormones. So glucokinase, hexokinase, or glucose 6-phosphatase in gluconeogenic pathways. And hexokinase basically, yes. Okay, so hexokinase and glucokinase is catalyze the same reaction, but just because these two enzymes have different tissue distribution. For example, we're just going to talk about the hexokinase is predominant in skeletal muscle, right? And glucokinase is more abundant in the liver, right? So because of tissue distribution, and also you try to connect the glucose transporters, like liver, mostly group 2, right, and skeletal muscle group 4. So these two different enzymes, they correlated with, different glucose transporters, just depend on which tissues, right? So something just correlate with, connect with tissues and glucose transporters, and so again, the hexokinase, because it's abundant in skeletal muscle, so usually coordinately work together with the group 4, because the group 4 is also highly expressed in the muscle, right? And work together and to control glucose enter cells, muscle cells, and to catalyze the first reaction of glucose, glycolysis in the skeletal muscle. And glucokinase is predominant in the liver and pancreas and correlated with the group 2, right, and regulates glucose uptake and metabolized through the glycolysis in the liver and the pancreas. And also these two enzymes regulate by different substrate, but I don't think you will need to focus on too much, particularly, not try to focus on too much on hexokinase, but glucokinase, right, in this. So, and when we talk about the glucose kinase regulation, so you try to remember the substrates, the glucose, fructose 1-phosphate, fructose 6-phosphate, glucose are the main substrates, right? 3 main substrates that regulates glucokinase activities in the liver. And also you wanted to remember the hormone, right? Insulin. So basically, you remember the regulation, right? The glucokinase basically is sequestered in the nucleus by the glucose kinase, regulates proteins. So fructose 1-phosphate is the substrate that can dissociate the glucokinase from glucokinase regular protein. So it's able to allow glucokinase release from nucleus into the cytosol for its action, right, in the cytosol, because the glycolytic pathway occurs in the cytosol. So in general, fructose 1-phosphate is activate glucokinase, right? And fructose 6-phosphate inhibits the release of glucokinase, so it actually inhibits the glucokinase activities. And insulin just directly regulates glucokinase activities or the transcription, gene expression. Yeah, so I think you can just remember the three substrate and two substrate, fructose 1-phosphate, and glucose are the activators of a glucokinase, right? And fructose 6-phosphate is inhibitors of glucokinase, and hormone insulin basically regulates or stimulates glucokinase. And also try to remember the mechanism, it's released from the glucokinase regulate protein in the nucleus, right, to the cytosol, so this process. And glucose 6-phosphatase is relatively simple compared to glucokinase. So insulin basically just suppress the glucose 6-phosphatase, right? So insulin, which probably is also easier, if you're thinking that way, the insulin should be inhibits gluconeogenesis, right? So this glucose 6-phosphatase is the key enzyme in gluconeogenesis. So insulin should inhibits glucose 6-phosphatase. We don't talk about much mechanism, so just remember, insulin inhibits glucose 6-phosphatase. Yeah, so this here he just put like two different pathway, glycolysis and gluconeogenesis together. So insulin stimulates glycolysis by stimulating glucokinase and inhibit gluconeogenesis and inhibit glucose 6-phosphatase. And the second pair of the key reaction, glycolysis and gluconeogenesis, and this two enzymes can be regulated by by functional enzymes, right? So I think this probably a little bit complicated. This pair of enzymes in gluconeogenesis and glycolysis is regulated by insulin or glucagon, but not directly by insulin glucagon. That indirectly, by insulin and glucagon, and this regulation involved in the bifunctional enzyme and a very important key substrate, fructose 2,6-biphosphate. Remember this? So, so here is sort of like the whole pictures how these two key enzymes in glycolysis and gluconeogenesis is regulated by insulin glucagon that involved in the bifunctional enzyme and key substrates, right? So fructose 2,6-biphosphate is a key substrate, but it is not directly involved in either glycolysis or gluconeogenesis. I mean, not the directly intermediates of glycolysis or gluconeogenesis, right? Does that make sense? But this substrate can directly regulate these two key enzyme activities, right? And the concentration of fructose 2,6-biphosphate itself, and the concentration of this substrate is regulated by functional enzyme. And try not to be confused this bifunctional enzyme to the key enzyme in glycolysis and gluconeogenesis. These are the two different sets of enzymes, right? So bifunctional enzyme basically has a two enzyme complex, phosphate, phosphofructose 2 kinase and fructose 2,6-biphosphatase, right? And these two enzymes work together to control or to regulate production of fructose 2,6-biphosphate, right? And bifunctional enzyme activity is directly controlled or regulated by insulin and glucagon, right? So you try to remember this sequential, events, how insulin glucagon regulates the key enzyme. First of all, insulin glucagon regulated by functional enzyme, and then control the key substrate production, and it is this key substrate that directly regulates the key enzyme in gluconeogenesis. or glycolysis, right? And this just show you guys that the how these two bifunctional enzyme control the synthesis or breakdown of this key substrate, right? And two key enzymes, and they do different things, right? For example, the phosphate fructose 2 kinase basically catalyzes the production of the fructose 2,6-biphosphate. So if this enzyme activity increase, you will expect to have high levels of fructose 2,6-biphosphate, right? And the other bifunctional enzyme, fructose 2,6-biphosphatase, and this enzyme, what this enzyme does is to break down this key substrate so that reduce the levels of fructose 2,6-biphosphate, right? And it's just again, this enzyme kinase catalyze synthesis of substrate, and phosphatase catalyze breakdown of substrate. And insulin also does the opposite to these two two bifunctional enzymes. So insulin basically stimulates kinase and inhibits phosphatase, right? And the consequences of this inhibition or stimulation of insulin leads to the increased production of this key substrate. And glucagon, that's opposite, inhibits kinase and stimulates phosphatase, right? And break down the substrate. So insulin in general increases the production of fructose 2,6-biphosphate, and glucagon reduces the production of fructose 2,6-phosphate, right? And then we see how this substrate regulates these two key enzymes in the glycolysis and gluconeogenesis pathway, right? So if increased substrate concentration, key substrate concentration, which under the fat condition stimulated by insulin, then this substrate can stimulate kinase, phosphofructose 1 kinase, the key enzyme in glycolysis, right? And in the meantime, this substrate can inhibit the fructose 1,6 biphosphatase, and this is a key enzyme in the gluconeogenesis. So this just sort of the complex regulator mechanism, to see how insulin that first of all regulates the key substrate concentration, and then control the key enzymes in gluconeogenesis and glycolysis, right? And similar, mechanism for glucagon, and just as opposite regulation, right? And the last key, no, not the last, but another key set of key enzyme, in the glycolysis gluconeogenesis, that pyruvate kinase, or the PEPCK, the PEPCK, or pyruvate carboxylase in the gluconeogenesis, right? And pyruvate kinase basically is stimulated by insulin and fructose 1,6 biphosphatase, and inhibits by glucagon. And PEPCK is the only enzyme that is an enzyme that only regulates by gene expression, does not involve in like phosphorylation or dephosphorylation mechanism, right, or allostatic mechanism. So the only way that control PEPCK activity is to control its transcription, so control the levels of PEPCK, right, gene expression. And glycogen, what you need to know is just the two key pathways in the glycogen metabolism, synthesis and the breakdown, right? And again, the liver and muscle are the key tissues that store glycogens. And the contribution to the blood glucose is different, right? In liver and the muscle. Liver is able to break down glycogen and release glucose into circulation during fasting, and muscle is able to break down glycogen but not able to release glucose. So the glycogen breakdown, usually just used as energy inside of the muscle cells. And glycogen synthesis involved in the several enzymes, right, and also some kind of molecules like UDP is a carrier of a glucose for glycogen synthesis, and also glycogenin is the primers, the key molecules, that protein that involve the initiation of a glycogen synthesis, right? It's important for the synthesis of glycogen primers. So glycogen primers contain the glycogenin proteins and plus the 8 glucose molecules, right? Of a linear chain attached to the glycogenin molecules. And the glycogen breakdown also involved several enzymes. And what you need to remember is a key enzyme in each pathway: the glycogen synthesis in the glycogen synthesis pathway, and glycogen phosphorylase in the glycogen breakdown pathway, right? These are 2 key enzymes in this pathways, and glycogen, we already talked about it. And the pathways, again, you just try to remember the key enzyme, right? And also, this is sort of a negative feedback regulatory mechanism. When we have a lot of glucose phosphate, a glucose 6-phosphate and glucose 6-phosphate and glucose, these 2 are the key substrates that can inhibit glycogen phosphorylase, to inhibit glycogen breakdown, right? And in the fat conditions, then when insulin levels high and glucagon levels low, and promotes the biosynthesis of glycogen, right? And so the glycogen synthesis activity can be activated by insulin, and then together with glycogen glucokinase, right? And for the glycogen synthesis. And in the fasting conditions, the glycogen phosphorylase activity is increased, right? So I think it's just it probably would be easier to connect all the physiological conditions and which hormones that responsible for which physiological conditions, and then the pathway. So go this direction, probably would be easier to memorize some things. For example, in the fat condition, we always have a high insulin level, low glucagon, right? And insulin should stimulate some, glycogen should be synthesized in the fat condition, right? And glycolysis should be active. So you can just thinking that insulin should stimulate key enzymes in glycolysis and key enzymes in glycogen synthesis, and glucagon should do the opposite, right? And during the fasting, you connect with the glucagon, high glucagon, low insulin, and then the gluconeogenesis pathway should be up, and glycogen breakdown pathway should be also increased. So the enzyme in these 2 key pathways should be upregulated by glycogen, and should be inhibited by insulin, right? So just try to connect all that together. It should be easier to remember. And for the regulation, I want you guys to remember cyclo-AMP. Right? So cyclo-AMP levels directly regulates the glycogen synthesis and glycogen phosphorylase. And during the fasting, when glucagon and epinephrine levels increase and cyclo-AMP levels increase, right? So what you can take a guess. So high levels of cyclo-AMP should stimulate glycogen breakdown, right? So, and should inhibit glycogen synthesis. So again, you connect with the hormones and fasting. And again, cyclo-AMP levels is increased by glucagon, epinephrine during fasting. And so high levels of cyclo-AMP should stimulate glycogen phosphorylase and inhibits glycogen synthesis. Right? And insulin just does the opposite. So insulin reduces cyclo-AMP levels, right? So reduces glycogen phosphorylase and stimulate glycogen synthesis activity. Yeah, it's just more information. So, this one. Yes. So if you go a little bit detail, how insulin reduces cyclo-AMP, and the mechanism as insulin inhibits the cyclic nucleotide phosphodiesterase, and this is the enzyme that catalyzes the formation of cyclo-AMP, right? So once this enzyme is down, then you have a less cyclo-AMP is formed, right? So this is sort of mechanism, how insulin reduces the cyclo-AMP level. Okay, so and this just kind of connection, I just talk about it. Glucagon, epinephrine levels increase during fasting, can regulate cyclo-AMP levels, and cyclo-AMP levels increase can stimulate protein kinase A, and protein kinase A can phosphorylate the phosphorylase, and once phosphorylase gets phosphorylated, it becomes active, right? So this involve more kind of mechanism, right? Protein kinase A phosphorylate phosphorylase and activate phosphorylase, right? And promote glycogen degradation. So this, I think, kind of comparison of fat fasting conditions and which hormones secreted and how glycogen synthesis and glycogen phosphorylase is regulated, right? Again, you try to connect this physiological condition and to remember, insulin is up in the fat condition, right? So glycogen synthesis should be increased, and glycogen phosphorylase should be decreased, right? And should promote glycogen store. And the fasting condition, glucagon, epinephrine levels increase and decrease, and reduces glycogen synthesis and stimulate glycogen phosphorylase, right? And glycogen degradation. And other regulating mechanism called allosteric regulation. So for glycogen metabolism, ATP, glucose 6-phosphate, and free glucose regulates phosphorylase, right? So phosphorylase, glycogen phosphorylase can be regulated by these three substrates. So glucose 6-phosphate and free glucose is inhibits, right? Remember the phosphorylase, glycogen phosphorylase, so inhibits glycogen breakdown. And the other activators, the 5'-AMP is also important regulators of phosphorylase, glycogen phosphorylase, and it's only in the muscle tissues. right? Because the muscle, in muscle tissue, this enzyme isoform has a AMP binding site, so be able to allow the 5' AMP to bind its binding site and to regulate the enzyme activities, right? So again, only occurs in the muscle, right? This regulation. And this is the last slide. I think you wanted to make a comparison, particularly focusing on the regulators or hormones, and the the substrate is different in terms of regulation of glycogen in between liver and muscle. And insulin, glucagon, and epinephrine were all able to regulate glycogen metabolism in the liver, but in skeletal muscle, glycogen is not able to regulate glycogen metabolism in the skeletal muscle because of lack of receptors, right? And for the substrate, for liver, ATP, glucose 6-phosphate, and free glucose are the regulators. And the muscle, free glucose is not the regulators for muscle glycogen metabolism, but 5' AMP is the regulators, right, in the muscle but not in the liver. And also lastly, I mean, we mentioned a lot of times, many times, you know, the liver contributed free glucose, right, to the circulation, and muscle does not. Again, the reason is just lack of glucose 6-phosphate. So in the muscle, glycogen will break down to glucose 1 or 6-phosphate, and then enter the the glycolysis pathways for further metabolism, right? Okay, so I think finished the review, and you guys can ask me questions when. Yes. From glycolysis, you mean, right? Oh, from TCA cycles. Okay, so TCA cycles, let me go back to the TCA cycles. So one ATP is produced at the substrate phosphorylation level, right? And then and then the rest. Let's see. And the rest ATP should be from NADH and FADH. So you go to the TCA cycles and see how many NADH, FADH is produced, and then you calculate, the ATPs. So in general, from the one molecules of glucose, the complete oxidized produce 36 to 38, right? ATPs, that including ATPs from glycolysis and TCA cycles. All right. Does anyone knows exactly? Yeah. I'm sorry. Oh, you have different question. Okay, yeah. How glucose 6-phosphate, right? Glucose 6-phosphate. Glucose 1-phosphate, you mean? Oh, fructose 1-phosphate regulates glucokinase. Did I say glucose? No, fructose. No, glucose 6-phosphate. Glucose 6-phosphate regulates the glycogen breakdown, right? Is that? Oh, Did I say? Yeah. Yeah. So let me see. I can't remember what I talked. Maybe it's. Um, Oh, okay. Yes, yes. You're right. I think the similar mechanism, like so glycogen breakdown is catalyzed by the key enzyme, right? Glycogen phosphorylase, and glycogen phosphorylase we know is inhibited by glucose 6-phosphate, and it's also inhibited by fructose 1-phosphate. You're right. Yes. So both substrate fructose 1-phosphate and the glucose 6-phosphate are able to inhibit glycogen phosphorylase and inhibits glycogen breakdown, right? Yes. Oh, that's a good question. So we're going to talk about the very late, I think, in the semester, like fuel utilization. Yes. So we talk about the sequential or timing. So usually the glycogen breakdown happen earlier than the gluconeogenesis. So always like we try to use the stored glycogen. If not enough, then we make the glucose. Yes. Yeah. Sorry. Oh, composition exam one. Yes, I think we have a 5 choose 4 selection, and then 15. Sorry, 15, right? Questions, and then the rest of question are multiple choice. So we have a total 55 questions. So mean 45 multiple choice. Yeah. Yes. Oh, you mean the TC cycles? Yeah, 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 sure. Let's see. Oh, I don't know for some reason. Okay. So TC cycles, the total, I mean, plus the, see the ATPs, this one here, right? It's directly generated in the TC cycle at the sort of substrate phosphorylation level, right? And then you see 1, 2, 3 NADH and 1 FADH, right? So you calculate it, and because this all in the mitochondria, so each NADH will generate 3 ATPs, right? So 3 times 3, 9 ATPs from 3 molecules of NADH, and 2 from FADH, so 11, 11 plus ATP, 1 ATP, so 12. So 12 ATPs are produced from TC cycle directly. Sorry, not directly. I mean the overall, right? I mean directly only 1 ATP, but indirectly from this, equivalent, reducing equivalence, right? So 12 ATPs generated from TC cycles, one at the substrate phosphorylation level, right, and the rest just from electron support chain, initially from 3 NADH and 1 FADH. Yes. Correct. Yes. So it will be 24. Yes. Thank you for correcting this. So it will be 24, right? One cycle you generate 12, and because you have a 2 acetyl-CoA generated from glucose, right, enter TC cycle, so 24. And 24 plus the 6 to 8 from glycolysis, right? And then, okay, one more place that also produces NADH is remember the pyruvate converted to acetyl-CoA, right? Also generate one NADH, right? Remember? So you also need to plus that, ATPs from the pyruvate dehydrogenase. So the total ATP will be 36 to 38. But that makes sense, right? 24 from TC cycle, 6 to 8 from glycolysis, and 3, well, you also time 2, right? 6 from the hydrogen, pyruvate dehydrogenase reaction, right? Right? Okay. Yes. Which pathway? In all the different pathways? Yeah, I mean, gluconeogenesis, the glycolysis is 3 pathway, right? I mean, 3 reactions, 3 key enzymes, and then glucokinase or hexokinase in the first reaction, and then phosphofructose 1 kinase, right, in the second key reactions in the glycolysis, and then pyruvate kinase. Yes. All right, so any other questions? Yes.
Going back to
Mm-hmm.
glycerol-3 phosphate shuttle, what 2 pathways was that connecting again?
Oh, you mean glycerol 3 shuttle and merely aspartate shuttle? Okay. Yeah, that is. So remember the NADH produced from the glycolysis in the cytosol, right? And because this cytosol, the NADH cannot pass through the mitochondrial membrane and go into, for oxidative phosphorylation to generate ATPs. So that means we need another molecules like glycerol 3 phosphate or malate, and these 2 molecules are able to pass through the membrane. So the electron that present in the NADH, first of all, need to transfer to the glycerol 3 phosphate or malate molecules, and then these 2 molecules carry the proton or electron from NADH and then pass through the mitochondrial membrane and enter the mitochondria, and then transfer the electron that initially from NADH in the cytosol to either FAD for if it's a glycerol 3 phosphate, right, or to NAD and generate FAD or NAD. So because of this, shuttle system, transfer electron to the different to generate different reducing equivalent in the mitochondria. So that's why, glycerol 3 phosphate shuttle carry electron and generate only 2 ATPs, and malate is able to generate, the shuttle is able to generate 3 ATPs, right? Yeah. Yeah. Mm-hmm. You mean group 4 deficient or something like this? Okay, so yeah, good question. So her question is if group 4 deficient will affect the insulin levels. Group 4 deficiency, actually you're not able to transport the glucose into the into the cells, right? So you raise the blood glucose level, which is very common in type 2 diabetes. And actually, in type 2 diabetes, the insulin levels increase, but it's not very clear, how what actually causing the insulin levels increase. The mechanism is not very clear, but it does. Yeah, actually, in the type 2 diabetes. Yeah. Oh, okay. I mean, depend on the cell types. Some cell types, they only have this shuttle system, glucose 6-3, glycerol 3 shuttle system, and the other type of cells has, the Mealy shuttle system, depend on cell types. So I can't exactly remember that. I think the brain is a cell type that has probably Mealy shuttles, and the other probably muscle has the glycerol 3 phosphate shuttles. The muscle cell. So it will be so in the muscle, for example, if it has a glycerol 3 phosphate shuttle, so the NADH produced from glycolysis will generate 2 ATPs, right? Or in other words, if we oxidize glucose in the skeletal muscle, we will generate 33 ATPs in the muscle, but 38 in the brain because of a different, the shuttle system. Does that make sense? Okay, yeah. You have, oh, okay. Quizzes, answers? Yeah, I can do that. Yeah. Yeah. Okay, sure, yes. So for gluconeogenesis, I guess would be better to go that slide. If precursors are lactate or pyruvate, then will involve the four reactions, 2 in the mitochondria and 2 in the cytosol, right? And if glutamate is the precursors and only involve one reaction in the mitochondria, and 2 reactions in the cytosol. And glycerol, there are just only 2 reactions in the cytosol, right? Okay. Yes, yes. Mm-hmm. Yeah. Any other questions? Yeah, Karen. Oh, sorry, I forgot about this. Yes, so do you guys have, let me see, I think I have maybe. don't have the. So I think the probably have two parts, right? I think we have posted like example. So the first part would be just answering the questions. For example, if you think you were including like 5 different papers or maybe more than 5, but you wanted to list the main, the major papers. For example, you think 5 or maybe 3 or 5 paper you think is the most important paper that you wanted to, focusing on. Then you answer the questions, right, that we have on that table. For example, what type of so you just list the paper by paper. So for example, in the first paper, it was like human studies, and second paper, animal studies, and you try to answer the questions, what type of study design that paper talk about it, and like, sample size or some other questions, right? You don't have to list for 10 paper if you have select 10 paper for your paper, but just list again 3 to 5 main papers, right? And answer that question. And then the second, I think the most important part would be like abstract, the exactly actual abstract that for your papers. So I will expect you guys to read all 10 papers if you select for your papers, and read all the papers and be able to, to write an abstract for your review papers, review papers. Does that make sense? And you also give us the kind of reference for your paper. Your question, yeah. How many? What? How many resources? Pretty much all of the reference that you wanted to include. Pretty much close to finish. Yeah. 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 I think we can actually postpone the due date since you guys have an exam. So what is the date next due date? What is the due date that we have? 11. We can postpone like 13 or I don't know. What is it? Next Thursday. Well, 14. Let's say Friday. How about next Friday? We can give you guys, extra maybe 3 days to work on it. Right? So, any other questions? I will send out email to you guys. Sorry, I forgot about sending this. Yeah, you wanted to including the 10 reference. We have like a 3-part, right? First, the table, you finish the question, and then the real abstract, and then the paper. The reference. Okay.